Our next speaker, our second speaker and last speaker of the day today is Woody Sherman. Woody is the Chief Innovation Officer of Psivant Therapeutics. Prior to Psivant, he was Chief Computational Scientist at Roivant Sciences, Chief Scientific Officer at Silicon Therapeutics, and Global Head of Applications Science at Schrodinger. Woody is also an Adjunct Professor at the University of Massachusetts at Amherst, where he gives lectures on computational chemistry, drug design, innovation, and company creation. Woody, after completing his PhD at MIT, has published over 100 peer-reviewed papers, including molecular dynamics, quantum mechanics, free energy simulations, and other areas of cheminformatics and machine learning. Woody's topic of presentation is From Concept to Clinic, discovery of a non-CDN sting agonist using QM, MD, and ML. Woody, the floor is all yours.
Great. Thank you for the introduction, and thank you for the invitation to be here and from the organizers. Just a quick question, you're seeing the full screen mode and not my little presentation mode?
Yes. Excellent.
Great. So I'm going to start off just with a high-level summary of our view on the importance of dynamics. I know this is the AI symposium, and we'll touch on AI as well. John did a great job at emphasizing the importance of dynamics as well, so I'll be able to get through this pretty quickly. Talk a little bit about Psivant Therapeutics and the computational platform that we've been developing since the days of Silicon Therapeutics through Roivant and continue to develop within Psivant. And then I'll focus primarily on the discovery of a non-cyclic dinucleotide small molecule sting agonist that's currently in the clinic. So Richard Feynman, over 60 years ago, said what we're still talking about today, that living things can be understood in terms of the jiggling and wiggling of atoms. So we've known this for a long time. The challenge, of course, is being able to make predictions and run simulations at scale where we can influence our drug discovery processes. So we start with the fundamental view that dynamics govern biology, and then take a flexible approach to solve problems as they arise. We focus on physics or ML as the challenge depends, as well as a fully integrated lab. Sometimes computation is the right tool to use, and sometimes it's not. And so being practical and focusing on what matters in the context of our drug discovery projects. When we talk about molecular dynamics, we're not talking about just running simulations and watching things jiggle around. We're actually primarily interested in free energy landscapes, the difference between different conformations and how those conformations relate to biology. And we don't do this only with molecular dynamics. We also integrate experimental data, and we feel that at least for the foreseeable future, that is going to be the most effective way of making an impact with molecular simulations. So why does motion matter, and how do we think about this? Well, we all know proteins are flexible, and that flexibility ends up influencing biology. And so how we handle these are with all-atom molecular dynamics simulations. And so we deal with waters and cofactors and ions and all the things that are in there. But our ultimate goal, as we peel that away, is to understand how molecules bind to proteins, and not just how they bind in terms of binding affinities, but also what happens when they bind to the protein, conformational changes as molecules lock proteins into certain conformations, and then how we can go about designing better molecules that both bind more tightly and have better profiles, but also influence the protein in ways that influence biology. And so we use these MD simulations to understand molecular mechanisms, which of course are based on dynamics, to predict pockets and binding pockets. Having a molecule that binds and sticks to a protein is insufficient. You have to understand not just bindability, but druggability. How do those pockets relate to function? That's also based on dynamics. And then, as John described, binding free energy calculations, which I believe are the most accurate in silico approach to predicting binding affinity, are also done with molecular dynamics, just as an example. So why not AI and machine learning for everything? It's been touched on a few times here, and more broadly, right now we're in a kind of data poor regime. We obviously need to work on that. I think it's a great place for NCATs and other governmental organizations to pitch in. It's been mentioned the PDB and how much has went into that and how important that is in the context of protein structure prediction. But that's just one of many areas that we want to solve. So there's right now currently poor data, a few insights as well. We're not at a stage where computers design molecules and make drugs. They are told what to do by people, and so you need to provide insights to people. And right now, there's not a lot in that space, although it's a great area of research that folks are working on. And there's just not a lot of end-to-end solutions. And I think several folks here have already talked about building along that process. But we're still early in the days. That's not to say that AI and ML are not useful. AlphaFold obviously has made a big impact, essentially solves the protein structure prediction problem, but without compounds. And as such, this is currently comparable to solving an apocrystal structure in a sense. And while that has some value, it's of minimal it makes a minimal impact in the context of drug discovery. We really need to be talking about how molecules influence, bind to proteins and can influence the structure. There are things like generative modeling, for example, that have been talked about, and tools that are out there that are quite useful, in the context of a broader suite of things. Active learning, Connor developed this tool called MolePal, but there's others out there as well, and we're big believers in active learning. It improves the efficiency of whatever you're applying the scoring function in. So whether that's docking or free energy calculations, it doesn't make the calculations more accurate. It just allows you to get through more molecules more efficiently, but still, that's of course very valuable. So overall, there are great tools out there. The field is evolving, which is great to see, and we're kind of in a fastly growing age with the investments that are coming in, but still relatively limited predictive ability, especially for ligand binding. One of the questions earlier in the chat was related to ML methods, and yes, ML methods can predict ligand affinities when you already have a lot of data, but once you have a lot of data, it's less interesting to be predicting affinity. And so when you're getting into the most interesting projects, novel drug discovery problems, especially for challenging targets where there might not be a lot of chemical matter, physics is still what we rely on most heavily. But we do use ML models for active learning, as I mentioned, but also ADME predictions and some generative modeling as well. So our approach has been, since the beginning, since the Silicon Therapeutics days, is to build a flexible toolkit. In drug discovery, one thing that I've learned many times over the years is drug discovery is hard for many different reasons, and each drug discovery project tends to be challenging for different reasons. And so while there are some recurring themes like predicting binding free energies, as you'll see in the part I talk about STING, that was a very unique problem, even though we were trying to predict binding energies in a lot of the standard stuff, and I'll talk about that. And so rather than building a black box or a single kind of tool to solve one thing really well, we've built a flexible toolkit that is based on quantum mechanics, thermodynamics, molecular simulation, AI and machine learning, as well as a robust computing infrastructure. And we have published a lot of this work along the way. Fortunately, I don't need to talk too much about binding free energy methods. John Codera did an excellent job describing the importance of these. The Open Free Energy Consortium is excellent, and we're strong supporters of the open science movement in general, as I'll talk about in a bit. But for free energy calculations, we have built up our own internal infrastructure to do this. We build on tools that are out there when they exist. For example, starting with David Mobley's LOMAP tool for certain pieces of it. We've had to build a lot of stuff on our own and ultimately integrate this. And the reason that we've done this is that we can really run at scale, which is super important for asking big questions about not just looking at a handful of molecules, but hundreds or thousands of molecules on a routine basis. It also allows us to customize the tools to solve our problems. And I'll explain again in the STING project why that was so important. And we've also built out other functionality that, again, is about enabling humans. We don't want to just produce a number at the end of the day. Ideally, we produce additional insights as well. And so we've created this atomic free energy decomposition capability where you can extract from the free energy simulations atomic contributions. And of course, this is not rigorous, but it's useful. You can't break up free energies into atomic contributions because there's things like entropy in there, but we do it in a way that we found to be very useful. And so this molecule can be good by a predicted minus 3.39 kcal per mole. What's even more useful is why is it good? What are the things that you might not want to mess with? What are the parts of the ligand that should be changed? What are parts of the protein that maybe you're unhappy that help lead to some additional design insights? And in addition to binding free energy calculations, we've developed a lot in the context of what we call biologically relevant protein motion. So, not brute force simulations where we're just watching proteins fold and unfold. I think D.E. Shaw and the D.E. Shaw Research Group has done an excellent job with Anton building a computer that can run those simulations at scale, but most of us don't have that kind of compute capability, and we need to think more carefully about the problem we're trying to solve. And in the context of our work in drug discovery, that really comes down to path-based sampling or various enhanced sampling protocols where you can investigate specific degrees of freedom or combinations of degrees of freedom in the context of what matters in biology. And so this is a big focus of us, and this particular work here describes some of the tools that we've developed along that process. But computation is not enough at this point. Even with an Anton supercomputer, we still can't simulate everything at the time scale and resolution that we need. And so we also integrate biophysical data. We've developed a framework that allows us to integrate data that's not necessarily atomic resolution, but we can bring in information that allows us to create more accurate and faster simulations around what matters, what's happening actually in the experiment. And so, for example, hydrogen deuterium mass spec is a very powerful tool to understand what parts of a protein are getting blocked from solvent, for example, upon complexation, ligand binding, et cetera. It doesn't give you any information about where the atoms are. It just tells which residues are blocked. But we can integrate that essentially as an additional factor in the MD simulations, and that allows us to get simulations that are consistent with the experiment. They're not constrained only to the experiment, but again, we're kind of biasing the simulations in an intelligent way so that they are consistent with the experimental data. And there's a nice example that we published here a couple of years ago on looking at heterobifunctional degrader molecules, so molecules that bring two proteins together. These tend to be pretty flexible, and we were able to use this approach with molecular dynamics and hydrogen deuterium exchange mass spec to get accurate molecular representations, essentially at atomic crystal resolution, but with full dynamics as well, which was also actually able to explain some previously unexplainable data about specific linkers and how those are influential in the degradation process. It's important, again, to bring this back to people. We've developed an integrated design process where we start and end with humans making decisions. Humans are still thinking about hypotheses, looking at thermodynamic data like that binding free energy decomposition work that I showed and others. We ultimately then enumerate large chemical spaces. We tend to stick to synthesizable chemical spaces, so we're writing out chemical reactions with known building blocks to explore chemical space, triage that down based on drug-like properties, machine learning ADME predictions, ultimately getting into molecular dynamics and free energy calculations. But all of this running behind the scenes ultimately gets presented to people on the team through the SciVision data platform that we've developed specifically for the purpose of integrating humans and computers in a way that I think is quite novel and useful, so that the results that come out of here, everybody on the team gets a link. They can go into this interface. They can look at all the computational data in many different representations, make decisions, provide opinions. It's all collected in a database, so then we're ultimately deciding which molecules to make. We've captured everybody's feedback from biology, chemistry, biophysics, and computation. We can't do this all alone. We are big supporters, again, of the open science movement. We have an open science fellows program with some great people that have been involved over the years. And so a lot of thanks to these folks and the work that they're doing, as well as all the many other open science contributors that are not out there. And we are also doing our bit on that side as well. We're actually developing a new molecular dynamic simulation engine that's made to be more performant on modern GPUs. At this point, a standard molecular dynamic simulation with 50,000 atoms doesn't saturate a GPU. These GPUs are growing so fast for these machine learning workflows that we have to think a little bit more creatively about how to leverage them maximally with using molecular dynamics. And so Storm is our attempt of making this more scalable, more performant with a modern MD infrastructure, and this is open source and released under the MIT license. All right, so with that as background, I just want to jump right into the story from our Silicon Therapeutics days, the design of a systemically available small molecule STING agonist. So STING, otherwise known as stimulator of interferon genes, is a central regulator in the innate immune system. It's circled here in red. STING undergoes a large-scale conformational change from this blue structure to this purple structure when it binds endogenous cyclic dinucleotides, and so this is what activates the innate immune pathway. Our goal was to design a non-cyclic dinucleotide analog to get in, to design a true small molecule that's systemically available. There were a number of companies at the time, a few years back, who had taken cyclic dinucleotides and made analogs that improve the properties. And cyclic dinucleotides are large, they're polar, they get cleared out of the body very quickly. They're non-drug-like, and a lot of the work to make analogs of cyclic dinucleotides only slightly improved those properties. Clearance went from 5 minutes to 10 minutes, but still nothing that could be systemically delivered. And our feeling was that a true small molecule could make an impact here. So what I'm going to show is a quick vignette and animation that combines a bunch of different parts of the simulation, and then I'll break it down into the individual pieces. So first of all, again, we're running all atom molecular dynamics simulations shown here. I'm just going to peel back the water so we could look at the STING protein under the hood. And what we'll first see is the conformational change from the active open conformation shown here to when a cyclic dinucleotide is bound. This is from inactive to active, and what we want to do is have molecules that bind to this active conformation. We did this by designing a small molecule, not this big molecule here, but a small molecule that dimerizes on itself in the context of the binding site. This is a unique mechanism and why so many unique tools needed to be developed. Now, this was initially an idea. These initial molecules didn't have any detectable binding affinity to human STING, but by analyzing high energy waters, we were able to build out some initial potency. We then started optimizing the pi-pi stacking interaction of the molecule on itself. Again, this is quite a novel mechanism, nothing in the PDB that really does this, but we were able to get crystal structures establishing this and go through some quantum mechanical calculations to optimize this pi-pi stacking interaction. And then ultimately getting into binding free energy calculations where, again, using thermodynamics, in this case, water thermodynamics, for example, to guide where we wanted to go, exploring small synthesizable libraries, using free energy calculations. And over the course of this process, we identified some interesting, unique conformations. In this case, in orange is a conformation of the protein that led to a very tight binding molecule, but it actually didn't have a strong pathway stimulation. And so we had to kind of step back. This is a very iterative process, and ultimately design molecules that lock the protein into a highly active conformation that's shown here in purple. And once we were able to identify that chemical series, we then focused on optimizing the ADME properties to bring that molecule from a lead to a drug. And this molecule is currently in the clinic for oncology. So just to take us through a few of those steps that went on there, we started with an idea that actually came from the mouse STING protein, and there was a molecule that formed this pi-pi stacking interaction, but it had no detectable binding affinity or activity to human STING. And so we first just started doing some modeling and came up with this idea, and then we had to start optimizing the molecule. And we went through various stages of, as we mentioned, looking at the water thermodynamics, looking at the pi-pi stacking, free energy calculations. All of these required customized tools to be developed. There's nothing out of the box that allows you to look at molecules binding as a dimer to each other and interacting with the protein. So again, one of the big values of open tools, customizable tools that you can really own and have a development team that can get in there and make modifications as needed. The first bit was, again, the water thermodynamics, super important. There's some great tools out there. We worked with Tom Kurtzman in the early days of Silicon Therapeutics, Tom Kurtzman, the original developer of WaterMap, and then GIST to follow up on that. And so there's a great tool out there called GIST, Grid Inhomogeneous Solvation Theory, that gives you pictures like this. And with our initial hypothesis of where our molecule was buried deep in the pocket, it's pretty easy to just look at a picture and say, wow, if we could just get out there, we could gain some potency. And that, in fact, did end up working, red being the highest energy water patches. And once we got our initial detectable binders, we started getting into that optimization of pi-pi stacking interaction. And even with just small halogen and 1 to 2 atom changes, there's still many combinations of things that one might be able to do with these 8 potential sites of chemistry. And we enumerated all of these potential ideas and ran this functional group-based symmetry adapted perturbation theory, FSAP, and again required customization of a tool. We work very closely with Todd Martinez from Stanford, who is the developer of TerraChem, a very fast quantum mechanical tool that runs on GPUs. And through a collaboration with him as an SAB board member, we were able to develop this tool in essentially real time. And from the identification of the need, it was just a few weeks later that we had a tool to compute the strength of this pi-pi stacking interaction. And it ends up that actually correlates quite well with the activity that we were looking to build, and we were able to explore thousands of molecules virtually and synthesize only 10 to come up with a molecule that was substantially more potent than where we were. And then from there was a combination of predicting and improving on ADME properties while maintaining and improving binding affinity. This is actually a distribution that you're looking at on the right here, which is all of the 200 or so compounds that we made over the course of the project. And I like to show these distributions as opposed to correlation plots and RMS errors, because basically this is what you would want to see if you were at Vegas trying to make a bet on something. There's clearly predictive value here. The molecules that we predict to be better than the starting molecule are shown in the blue distribution, and on average they end up being better. And if you wanted to select from a pool of molecules, you would much prefer this blue distribution than a average distribution, a random distribution, or the red distribution where molecules are predicted to be bad. But there is overlap, and there are molecules that we predict to be bad that we ended up synthesizing and they were good. These are things that we would call false negatives. We'd say they were bad, they were really good. Why did we make these? We were probably trying to improve other properties beyond just binding. And then of course there were molecules that we predicted to be good that ended up being bad, false positives, but again, in general, highly predictive and very useful, and just some specific examples of the kind of types of perturbations that we were making on this molecule and the experimental binding energy versus our predicted RBFE energies. Just want to have a little side point here that binding's not always what matters. In this particular case, we were looking for stabilization of a particular conformation. So it ended up that experimentally in the lab, binding affinity, IC50, for example, on the y-axis here, didn't actually correlate with cell activity. It took a while for us to figure this out, but we ultimately came up with a surrogate assay, a DSF thermal stabilization assay that much more closely correlated with what we care ultimately about, which was, in this case, cellular activity. And also just wanted to mention that it's important to know what you're going after and what your various models are going to be throughout the process. One of the problems with that earlier STING molecule is that you could see that it does stabilize the mouse version of STING, this DMXAA molecule, but it had no activity on human. And so when we were going through this process, we were looking at the human wild type STING as well as this highly prevalent HAQ allele, as well as the various preclinical models. We want to make sure that the molecules that we have are also going to work in mouse models and the other models. The reason it matters for even the tox models is that the main concern with STING is on-target tox, not off-target, but on-target tox, specifically cytokine storm, for example. So we wanted to make sure that it was binding and doing what it was doing across the various relevant species. So ultimately, through this project, we were able to design a potent and selective small molecule STING agonist that worked on wild type STING, the HAQ, as well as the other variants, more potent than the cyclic nucleotide analogs. And ultimately, in the preclinical models, a single dose of this compound resulted in complete tumor elimination. This is, again, an innate immune-mediated mechanism. So the goal isn't necessarily to inhibit a protein or block something permanently. It's to turn on the immune system, turn these cold tumors hot, and then have the body take care of the rest. And so what we're showing here on the left is in dark, the untreated population, and in cyan is the treated population. And just a single dose resulted in complete tumor elimination. Perhaps even more interestingly, 3 months later, these perfectly healthy mice were re-inoculated with the tumor. And while the tumor grew in naive mice, the previously treated mice without any additional treatment, it didn't didn't grow. So it was some kind of innate to adaptive immune learning that went on in this process. So this is just a summary of the various different tools that I covered and the tools that we use, but the main conclusion is we explored millions of molecules virtually. We applied custom tools throughout the problem project, especially because of these unique problems of this homodimerization, conformational change, and ultimately got to our clinical molecule. So just in conclusion, drug discovery is hard. Each project's hard for different reasons, and my opinion is that black boxes are not quite there for drug discovery. We still need to have bespoke solutions to solve problems as they arise. That's why we've developed a toolkit approach, so you can apply the right tool at the right time, come prepared for anything and everything, and have not just the tools, but the people who can do additional development with those tools as necessary. The future obviously holds great promise for AI. There's a lot of great stuff coming out, but we live in the present right now, and we got to use what's there. And so we're investing in both directions, physics and AI, and we're very happy with the direction things are moving in the field. Computational approaches can add a lot of value right now. We did that at Silicon, we did it at Roivant, and we're continuing to do that at Psivant Therapeutics. And very importantly, computers alone can't solve this problem. It's really about the integration of the lab and the in silico approaches, and of course, with the appropriate people leveraging those tools to make decisions. So with that, I want to thank everybody, and I'm happy to answer questions.
Thank you so much for the excellent presentation, Woody. We'll take a couple of questions. I think we are almost there. I'll start with the bottom two questions, actually. They're almost similar. What made these molecules self-dimerize? Is the dimerization and structured process?
Yeah, so it's through a pi-pi stacking interaction. It probably is prevalent in solution to some extent, but due to entropy and the concentrations and stuff, it's not observed. There's actually another group after we did this that demonstrated that there is probably some very weak aggregation, dimerization that's taking place in solution, but it is through a pi-pi stacking interaction, and it's really the combination of the binding site and the fit to the binding site coupled with that pi-pi interaction that's driving things toward that final thermodynamic state, protein plus these two molecules together. So again, likely there is some favorable interaction in solution. What we're measuring here is just on their own, what is that dimerization energy? And it is a favorable pi-pi stacking interaction. But again, at concentrations in solution with entropy and everything, you're not going to be seeing this dimer. It looks, these molecules look and behave like monodispersed true small molecules in solution with solubility and permeability of a small molecule, but with some population very likely that's dimerized, and that's the form that's stabilizing the active conformation of sting.
Maybe you take one more question. You are using your own MD engine, as far as I understood. What kind of accurate force field do you use? Also, have you tried or plan to try creating own ML-based force field along the lines that John Codera outlined earlier?
Yes, absolutely. So we're very much keeping up with the field, doing our own development, also working with what's out there. So internally, we've developed a infrastructure that basically does bespoke parameterization, quantum mechanical torsion scans, quantum mechanical rest fitting of charges. This is kind of the state-of-the-art type of approach that open force field uses as well. Open force field wasn't a thing when we started at Silicon Therapeutics, and so we developed this on our own. Now, actually, we use the open force field quite regularly. We also are exploring some of these ML-based models. We're not developing those ourselves, but I think there's a lot of promise there.
Thank you.